There we go. Okay. Oh my God. Okay. So once again, hi everybody. Welcome to this review session. We're gonna go over some stuff that was gone over in class, I think, or well, the recorded videos since Wednesday, because Sebastian covered the first section of the week 1 material, and I'll go over week 2. Just a reminder, I think your problem set's due Sunday. Or a quiz or whatever. Some things due Sunday. But so hopefully this will help you kind of figure it out. All right, so this is just a quick overview of what we're going to talk about: environmental effects on protein folding, protein function regulation, enzymes, and protein stress response. And then we have this fun little graphic. Okay. So protein folding in the environment, I know from last section, I'm used to saying last week because when we had normal semesters, it wasn't as condensed as this one is, but the last section that we talked about was protein folding. And if you remember correctly, we talked about how polar amino acids are more likely to be on the outside, or hydrophilic amino acids, and hydrophobic amino acids are more likely to be clumped on the inside when a protein folds. This is known as the hydrophobic effect. But so when the protein folds, there's a lot of factors that play into how it folds and its function, including the environment. So the environment is why cells control for certain factors such as temperature, pH, and salt concentration. These are really important because they definitely affect how a protein is able to fold or not fold. Just some quick terminology: denaturation means protein unfolding, and then if you see renaturation, that means protein folding. So there are specific proteins in the cell called chaperones. These help proteins fold or help proteins restabilize if they're not folding correctly or if they're unfolding. So like to think of these as chaperones on a field trip. Chaperone proteins help proteins behave just like chaperones on a field trip help kids behave. They just make sure the proteins are folding correctly and not misfolding or clumping or anything that could be bad. So just a quick disclaimer. So for this class, we consider, because this will make more sense in the next slide, but for this class, we consider covalent bonds to be the strongest bonds between molecules. Most of the time, if you're asked about covalent bonds and environment, assume that the covalent bonds are too strong to break based on the environment. So this will make more sense in the next slide. So one thing in the environment that affects protein folding is temperature or thermal energy. Thermal energy affects the stability of non-covalent bonds. So this can affect like I said, this can affect the stability of covalent bonds too, but only in very, very extreme conditions that you probably won't find within the body. Like even boiling point won't really affect covalent bonds. So basically, anytime you hear like pH, salt concentration, or temperature, just think that covalent bonds are too strong to be altered by any of these environmental factors. So you probably won't see any questions like, what does temperature affect covalent bonds? And it doesn't really affect it in our bodies. So just a quick reminder, less thermal energy equals more stable bonds. So think of it as there's less like input from something that makes the bonds more crazy. So there's less thermal energy, those bonds are more stable. But just remember, a lot of times students fall into the trap that more stable is good. That's not always the case. So this is what we consider the Goldilocks hypothesis. You don't want too much, you don't want too little, you want it to be just right. So sometimes if there's too little thermal energy, the diffusion and motion in the environment may not be enough for interaction to occur, and that sometimes can't be good for the cell. So don't always think that more stable is good. Think of how our bodies have evolved to find conditions that are just right for our proteins and cells to function. So pH and salts. The pH and salt concentration in a cell can affect the ionic or charged interactions between molecules. An example, these are 2 examples here. So some amino acids, if you remember from the last part, that are more acidic will be negatively charged at a specific pH. Then that will affect their interactions. If they're negatively charged, they're going to interact more with positively charged other molecules or stuff like that. And then salt bridges and tertiary structures can be affected. So remember, disulfide bridges or salt bridges are only in tertiary structure, but those can be affected by the pH or salt concentrations. Another aspect of temperature that affects affects bonds and protein folding is not light energy. So light energy can introduce another form of energy to the system, kind of like thermal energy, but it's light energy. This also affects non-covalent bonds. Once again, remember, we're not really talking about covalent bonds here. And higher energy wavelengths can disrupt non-covalent bonds. So this is, you don't really need to know too much detail about this one. Just know that if you are hitting at it with certain energy wavelengths, it can disrupt non-covalent bonds, and think of like an X-ray or something like that can introduce a different form of energy to the bonds involved in protein folding. There are certain cases of light energy that can be extreme that affect covalent bonds, but once again, don't worry about it. Check that. Okay. Oops. Hey, computer, let's work. Thank you. Okay, good. Okay. 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 So talking about protein folding, let's talk about protein unfolding. So when proteins unfold, like I said before, they're hydrophobic R groups that were once tucked away on the inside, that hydrophobic effect. These are now exposed to the polar aqueous solution of the cell, and they don't like that. That's why they're called hydrophobic. They don't like water. So the hydrophobic R groups that are used to being on the inside are now going to scramble to bind to other hydrophobic R groups on other unfolded proteins nearby, because they don't want to bind to polar or any water solution. It's in the name, hydrophobic. So they want to bind to other hydrophobic R groups on other proteins, and this causes protein aggregation. So think about when you cook an egg, it gets more solid. That's because you are heating it to a temperature where the protein is able to unfold, and the hydrophobic R groups that are now exposed are scrambling to bind to other hydrophobic R groups, and that's what causes it aggregation. So, cooking an egg, that's good for us. We can eat the egg, whatever, but this protein aggregation can also cause neurodegenerative disorders. For example, Alzheimer's, which is believed to be caused by protein aggregates of tau or alpha amyloid proteins, and then mad cow disease, which is caused by misfolding of proteins called prions, which can actually be infectious. So those can be some examples of when protein aggregation is bad. But to combat this, cells have specific responses called stress responses for unfolded proteins. There's two that we're going to talk about in this class, the heat shock response and the unfolded protein response. So the main two things with these is, or just remember that the heat shock response occurs in the cytosol in the nucleus, and the unfolded protein response occurs in the ER. And we'll go a little bit more into detail, but there's a little diagram here of, this is when it's folded, all these hydrophobic R groups on the inside, and then now they're exposed, and now they're like, oh shit, what do I do? Okay, so then it's gotta, they're gonna scramble. So this is just quick on chaperones, and I kind of talked about it a little bit before, but they're just special proteins that are recruited to help other proteins fold or stabilize them. And most likely, it's the chaperone proteins that sense misfolding or unfolding. And here's a little diagram of protein synthesis, which leads to an unfolded protein, because when you make a protein, it's not going to come out folded, it has to fold itself. And it can either aggregate because it didn't fold properly, or the chaperone will assist and help the protein fold quicker and correctly. So we talked about the two, I mentioned the two responses. So the heat shock response is the first one we'll talk about. This is the one that occurs in the cytosol in the nucleus. I wish I had an easier way to help you remember that, like a mnemonic. If someone wants to come up with them, that'd be great, but I don't. Just try to remember that cytosol and nucleus happens in the heat, is the heat shock response. So basically what happens is the cell senses that something is wrong with a protein that is misfolded or unfolded. So what it does is it usually will tag them and ship them off to the proteasome where they will be destroyed. So during this process, chaperone production is upregulated or increased because it's going to need more help tagging these proteins and getting them either removed or folded properly, but usually what happens is they will be sent off to the proteasome. This is just a quick little diagram of what it looks like. So I know some of it doesn't really make that much sense, but basically what happens is it doesn't fold properly, it's sent to the proteasome, and the proteasome will degrade the proteins. So they can't aggregate and be harmful to the cell. The other response is the unfolded protein response, or the UPR. Oh, that actually might work. So the UPR occurs in the ER. Try to think of it like that. So for proteins that are going to the endomembrane system, which is something we'll talk about, I think, in the next section, talk about protein, like where certain proteins end up and stuff. So the endomembrane system, for right now, that you need to know, just includes the ER. So there's an ER specific stress response called the unfolded protein response. So cells sense the unfolded proteins in the ER. So these unfolded proteins are unfolding in the ER. And basically what happens is the ER responds by reducing the amount of proteins that it's translating or making, because it needs to focus on the current proteins that are messed up right now, so it can fix them and send them off so there's not damage to the cell. It also makes more chaperones and makes more space for proteins. So this is how they do this. So they decrease translation by reducing folding load in the ER, because like I said before, they want to focus specifically on the messed up proteins, or the unfolded proteins that need to be taken care of. They upregulate chaperone production, which will help refold and unfolded and misfolded proteins, like we talked about before. And then to make more space for these proteins, what the ER does is it increases lipid biosynthesis. We don't need to go into like the specific mechanism of how it does this, but basically when you increase lipid biosynthesis, you make more space in the ER for proteins. And then once these three steps occur, what happens is the proteins are retrotranslocated out of the ER to the proteasome. So that's a big step in the UPR, is that there are no proteasomes in the ER. The ER has to ship the proteins out of it so it can be sent to the proteasome when it's in the cytosol for degradation. And then here's just another quick little diagram. So basically, just think of this is where it starts. ER stress, you have a lot of misfolded or unfolded proteins. So what happens is it stops the increase. So your transcriptional response will increase in the ER protein folding capacity, and then the translational control of mRNA decay will decrease in ER protein folding and load. This is a little confusing, but basically just remember these three steps right here, and then these three occur in the ER, and then the proteins are sent out of the ER to the proteasome. So that's the UPR. Nikki, do you have any questions so far? Am I going too fast? Sometimes
no, you're good. Thank you.
Yeah, no problem. Okay, awesome. And anybody watching afterwards, if you have any questions, you can always email me. I'll put my email on the slide at the end again. Okay. All right. So now we're going to talk about protein ligand interactions and enzymes. Okay. So protein ligand interactions. Basically, you have your protein right here. Here's your binding site and a ligand. So a ligand is literally anything. We never really specify what it really is, but it's anything that will bind to a protein. So for basically, all you need to know for protein ligand interactions is that the number and strength of chemical bonds formed between the protein and a ligand determines the affinity. So basically the affinity is the strength of the interaction. A higher affinity equals a stronger interaction. So if these two have a high affinity, it's going to be harder to pull them apart. And that is dependent on the number and the strength of chemical bonds between these two molecules. Usually it's the higher number of bonds, the higher the affinity, and obviously the higher the strength, the higher the affinity. So to determine the affinity, we have what's called the KD, the dissociation constant. KD is the average rate measurement of how often a protein dissociates from its ligand. And to calculate it, you have the concentration of protein times the concentration of ligand divided by the concentration of the complex. So the complex is these two together. So it's basically this separate times this separate divided by how many of these together there are. There's a whole big mathematical way to calculate it. There's graphs, all this stuff, but that's the main idea you really need to know for this class. She's never going to have you calculate this equation, but you just need to know that the smaller the KD, so the value of KD, the higher the affinity or the stronger the interaction. So it's an inverse relationship. The larger the KD, so the larger the value of KD, the lower the affinity and the weaker the interaction. So if you have a small KD, say it's like 1, I actually don't know if that'd be small, but say you're comparing 2 KDs. This one's one is equal to 1 and the other is equal to 3. KD that's equal to 1 is going to mean that these two are harder to pull apart. KD equal to 3 means that they're easier to pull apart. So it's that inverse relationship. Sometimes people get confused. They think it's the opposite, but just try to remember that's inverse, okay? Okay. And please stop me if you have any questions at all. You can put in the chat, whatever. I actually don't know if I can see the chat when I'm sharing, but you can just yell out and be like, hey, go back, stop, okay. All right, so enzymes. I'm sure you've all have heard of enzymes before, but enzymes are biological catalysts. They increase the rate of a reaction by lowering the activation energy required for that reaction to occur. And by doing this, to lower the activation energy, they stabilize the transition state, which we'll talk about in the next slide, because there's that reaction coordinate graph, which I'm sure you've all seen many times. So they make and break covalent bonds. So this is important because up till this point, we've talked about non-covalent bonds, but enzymes make and break covalent bonds. So this means that they change the chemical makeup and structure of molecules. These are chemical reactions. So that's pretty important. And we'll talk about why they're needed in a second, but basically this is just the mechanism of how the enzymes work. So the substrate or the originating molecule enters the enzyme's active site. The enzyme changes shape slightly as it binds, and then the enzyme produces different products from the starting. But the main, a big point to remember is that the enzyme is never changed throughout the entire reaction. Only the substrate is, and the products are different from the substrate. But the enzyme is the same through every single step. So that remains unchanged throughout the reaction. So enzymes are needed because they speed up reactions that would happen on their own, but are too slow for the function of the cell. So an example is RAN gap, which you guys are probably like, what the heck is this? I've never heard of that before. But this is something we'll talk about, I think, in maybe 2 weeks or 1 week. I actually don't know. No, 2 weeks, 2 weeks. So RAN gap is a certain type of enzyme that does something important in the cell, but basically without an enzyme, its function would be far too slow, and it basically is like the cell doesn't even have that, it doesn't even have this like protein, because the enzyme needs to regulate and speed up the reaction. Otherwise, it just literally doesn't do anything in the cell. So that's one example, and that will make more sense when you guys see that actually occur in like a couple weeks. So enzymes can be regulated. And this is also really important because this means that the processes that the enzymes act on are also regulated. So say like this is a really important substrate and it needs to make these really important products. Say it's something about, I don't know, say it's like blood pressure. Say like by making these products, you'll lower blood pressure, right? It needs this enzyme to to make these products to lower blood pressure. If the cell, so say the body's like, oh, I have too many, but I already have really low blood pressure, it can stop this enzyme from functioning and then it won't create these products. So it's really important that enzymes themselves can be regulated, because then the entire process can be regulated. And it can be like, oh, I don't need these products, so it just shuts down this whole pathway. So that's really important. And then another thing that sometimes students forget is that not all enzymes are made of 100% protein. So most of them are, so most enzymes are proteins, but some are ribozymes, which means that they have an RNA/protein mixture. I don't have an example of a ribozyme for you, but they do exist. So this is that reaction coordinate. So remember that enzymes stabilize the transition state, which would be located somewhere around here. Like it's usually right here, it stabilizes it. So when you stabilize something, you're decreasing the energy of it. It makes it easier for this reaction to go over this hump because now it's lower. So yeah, more stable means less reactive or less energy. So this red line is without the enzyme. Oh my God, I need to stop doing that. it takes a lot more energy to get over this hump for products to occur. And then the blue lines with the enzyme. Just quick terminology. A spontaneous reaction is the energy. It occurs when the energy of the products is less than the reactant. So this is an example of a spontaneous reaction. A non-spontaneous reaction is when the energy of the products is more than the reactant. So it requires that energy is put in. In this case, energy is released because you have less energy in the products. So this is, yeah, an example. I don't think there's anything else I want to talk about on the slide. Do you guys have any questions? We good?
I'm good, thank you.
Good, awesome, cool. Okay. There was one thing, something about free energy. Nope, not gonna get into that because that's MCAT. Okay, so this is how enzymes catalyze chemical reactions. There's these 3 ways that Laura talks about a lot. And just remember, enzymes are highly regulated, like I said before. So A shows an example of an enzyme that binds 2 substrate molecules and kind of puts them in a way that they're closely interacting. So it encourages the reaction between them. So think of it like this red molecule originally without this enzyme was like way over here. And then this blue molecule is like way over here. So basically the enzyme just like grabs them both. Is like, okay, do your job, like work and make a reaction occur. So that just orients them so they're, close together and encourages reaction. B shows an example of how how an enzyme can rearrange the electrons in the substrate to create this partial negative and partial positive charges that will favor a reaction. The mechanism of this, I don't actually know the full mechanism, and I don't think you need to know it at all, but it just shows how the enzyme can change the charges on a substrate to encourage a reaction. Something about this might be that like these charges may make the activation energy much lower for a reaction to occur. And then C shows how an enzyme will strain the bound substrate molecule to force it towards a transition state to favor a reaction. Same with this one, kind of the same. It's one substrate, it's not two. And it just kind of strains it so that its energy is more favorable. So this quick question, I had this in, this is also an MCAT question. So if you can answer this, you're doing pretty well. If med school is something you want to do in your future, but don't worry also if you can't get it because it's literally from the MCAT. But it's just a little exercise. If during an enzyme catalyzed reaction and intermediate forms in which the substrate is covalently linked to the enzyme via a serine residue, can this occur at any serine residue or must it occur at a specific serine? What do you guys think?
I'm not really sure, but I would think like there would be like specificity where it would only occur at a specific site, but I'm not really sure.
Yeah, no, that's exactly right. Yeah, so it must occur at a specific serine residue because enzymes are very specific. That's why this highly regulated. But yeah, so good job. That's basically the answer. Enzymes are very, very specific. They will not just bind to any similar molecule. It will be very, very specific. So that's basically the main point of this question. Okay, so protein function regulation. We're going to talk about allostery. So Laura has mentioned time and time again, she does not use allostery as a term, but some people do, so you should know it. You should just know that allosteric binding means something is binding to a spot other than the active site on an enzyme, and that is a form of regulation. So if you look at this slide, so this is the active site right here. And then you can either have a substrate that can bind here. This green substrate looks like it could fit here, but then you can have something else bind at the allosteric site, which is located somewhere else on the enzyme. So in this reaction, this red cube will bind to this allosteric site, and what happens is the enzyme changes conformation. So this is an inhibitor. So now this active site is now changed and the substrate can't bind. So this is an example of allosteric inhibition. And then for allosteric activation, it's kind of the same thing. So this is this altered active site. Maybe it's this enzyme going from here. Now it needs to bind to this green half circle. So this activator will also bind to this allosteric site, and then it will change conformation of the enzyme. So the active site is now available. So this is an example of this main thing where structure equals function. You're going to hear that over and over and over in this class, structure equals function, because basically it is what it sounds like. The structure of the enzyme here determines its function. It can't bind to the substrate. Here, it can bind to the substrate because the structure changed. So that's really all you need to know about allosteric. It's not too complicated. It's just anywhere that's not the active site. And it's a form of regulation, as you can see here. So post-translational modifications occur after a protein is made to regulate function. They're typically reversible, and they're typically switch mechanisms. So this means that these post-translational modifications can either change the surface chemistry, change the structure, or change the function of the protein once they are added. Some examples are methylation, ubiquitination, and acetylation. So this is basically just how this happens. So the protein is translated, and then the cell signals for the direct addition of covalent modifications. So it can either be this yellow circle, this green shape, this blue triangle. And so yellow is phosphorylation, green is ubiquitin, and blue is acetylation. Phosphorylation is another example of post-translational modification. So what happens is the code is read, and then it can either bind to proteins Y and Z, so it's protein X, or it can move to the nucleus, or it can move to the proteasome for degradation, or it can move to the plasma membrane. So that is how the protein's regulated, by adding these modifications. So say by adding a phosphorylate, say by phosphorylating the protein, this tells the protein to move to the nucleus. Or if you add ubiquitin, this tells the protein to move to the proteasome for degradation. That's an example of ubiquitin-mediated proteolysis, which we'll talk about a little bit later in the class. So basically, these little tags that are added after the protein is translated, so CN to C, N-terminus, C-terminus, this causes the protein, it tells the protein basically what to do. They are also typically reversible. They're not usually, like if the cell decides it needs to reverse it, it typically can. But basically these are just signals that tell the cell or tell the protein what to do. So an example is P53, which is a tumor suppressor gene, which we also talk about a lot in the cell cycle later on in this class. But as you can see near the N-terminus, it has a lot of phosphorylation, and then it has like a variation of ubiquitin and acetylation at the end. But basically these are just tagged on to tell the protein like where to go and what to do. So that's an example of protein function regulation. And then the last example of protein function regulation is nucleotide binding and hydrolysis. So we talk a lot about GTPases and ATPases in this class. They're basically categories of enzymes that hydrolyze GTP and ATP respectively. It's never both, one or the other. So you never have a GTPase that hydrolyzes both GTP and ATP. It's only going to be GTP. So they use this activity to regulate activity. They hydrolyze a triphosphate to a diphosphate. That's what hydrolyzation means. So you're turning GTPases turn GTP to GDP, and ATPases turn ATP to ADP. It's common in signal transduction pathways, which we will talk a lot about in this class. So this is just a diagram of how this works. You have the GTP binding protein, you have GTP hydrolysis. So when it's in the GTP bound state, technically this enzyme is on and active. Then you have GTP hydrolysis, you lose a phosphate, so now you have GDP, and now it's inactive and off. And then this slowly will occur, that GDP diffuses out. Now you just have this GTPase with nothing in it. And then now GTP can quickly diffuse in and turn it back on again. So basically all you really need to know is that by binding GDP or GTP, this is an example of a GTPase, you can regulate the function of a protein or an enzyme by binding to it. So when it's in its GTP bound state, it's active and on, and when it's in a GDP bound state, it's inactive and off. And this is the same for ATPases most of the time. It's just with ATP and ADP instead of GTP. So that is all the material to go over for your next assignment that's due on Sunday. But before we do the practice questions, does anyone have any questions?
I'm good, thank you.
Good. Okay, cool. All right, so these are some practice problems that can kind of just correlate to what we just talked about. So number one, where in the cell is the unfolded protein response initiated?
Was that the endoplasmic reticulum?
Yep, yep. The UPR is in the ER. So yep, that's the in the endoplasmic reticulum. And the other one is the heat shock response, remember, and that occurs in the cytosol in the nucleus. Okay, so number two, which of the following ways are ways that the cell responds to unfolded or misfolded proteins? A, upregulating translation, B, increase chaperone production, C, translocate proteins to the ER, or D, increase lipid biosynthesis.
Would it be A and D?
A and D. Yep. So yeah, so you would not want to. Oh wait, nope. Sorry, I'm sorry. That's not.
Sorry,
not A and
sorry,
D.
B,
B. Yep.
B and D. Sorry,
Yep. B
yeah.
and D. Yeah. Sorry. Yeah. So you would not want to upregulate translation. You want to downregulate it because you don't want more proteins if you're trying to fix the ones you already have. You do want to increase chaperone production because you want more chaperones to help fold the proteins properly. You don't want to translocate proteins to the ER. You'd want to translocate them to the proteasome or the cytosol so they could go to the proteasome because there's no proteasome in the ER. And you would want to increase lipid biosynthesis because that will increase space in the ER for more proteins. Okay, so now in 3 and 4, a little bit more about the last unit. What effect would a molecule that disrupts hydrogen bonds, such as urea, have on protein structure? So which level of protein structure would urea disrupt?
Both secondary and tertiary and quaternary, I guess, if it has it.
Yeah. So exactly. So it would not affect primary structure because there's no hydrogen bonds in primary structure. It's just the amino acids, but it definitely would affect secondary. And then if there's a quaternary structure, it would. And it kind of is like a trickle down thing. So if it affects secondary, it's probably going to affect tertiary and quaternary if it has quaternary. But the main idea is it would definitely affect secondary because that's where hydrogen bonds are usually involved. So awesome. And number 4, if you mutate your protein of interest at the 21st amino acid, have you mutated an amino acid closest to the N terminus or the C terminus? What do you think?
I got this wrong on the practice problems, but I think it said you would have to know how many amino acids are in the protein.
Exactly, yeah. So the average protein, so most proteins have at least 100 amino acids or more. You're really not going to find ones with less than 100 unless she specifies. So says she's like, oh, your protein of interest has 22 amino acids. Then it would be their C terminus would be the answer. But because it's not specified, you can assume that a protein has at least 100 amino acids or more. So in this case, it would be the N terminus because it's closer to 21 is closer to the N terminus than 100. But unless she specifies and tells you the exact number or says that you can assume that there's 100 or more, that's how you would answer this type of question. Does that make sense?
Yeah, thank you.
Yeah, okay, awesome. Yeah, so that's all I have. I'll put my email back up. It's r.bolognese, like the sauce, at umass.edu. If you guys have any questions, you can obviously email me. But yeah, that's all I have for now.
Thank you so much.
Yeah, no problem.
Have a good night.
Thanks, you too.